Good morning, everyone. I'm Dr. Ted from IMU University. Today I will be presenting our group work on role of interleukin 6/8/10 tumor necrosis factor expression in gastric cancer, which is a hospital-based case control study. These are my research team. So as we all know, gastric cancer, although it is not listed as top 10 gastric cancer for Malaysia in terms of mortality and incidence, but it is still the leading cause of newly diagnosed gastric cancer globally and leading cause of cancer-related death, according to the report. So the main issue of gastric cancer is always late diagnosis and usually at the advanced stage. So as we all know, it has strong association with diet, lifestyle, and ethnicity, geographic location as well. Our aim is to observe between the expression of these cytokines in gastric cancer cases and control from Hospital Tunku Jafar, Negeri Sembilan, Malaysia. That was our method. First, we compiled the data from 2005 to 2018 from clinical pathological to the clinical pathological analysis. And first, we did from the FFPE samples, we did the detection of Helicobacter pylori using the PCR. And then secondly, we did the gene expression analysis and genotyping done. Genotyping will be done. I'm saying that will be done because this study is still an ongoing study. By the time I report this abstract to the conference, genotyping has not been completed. So far, our result is, first I would like to present the ethnicity incidence changes in ethnicity. According to the previous literature, Malaysian Chinese have the highest population, followed by Malaysian Indians and Malaysian Malay population. But now from our observation, Malay population is becoming the second highest incidence. And then youngest was the 19-year-old gentleman, Malaysian Indian gentleman, oldest was the 93-year-old Malaysian Chinese women. So another significant finding is H. pylori detection in gastric cancer patient, as we all can see in this. H. pylori detection is lower than lower population, H. pylori negative in gastric cancer, and H. pylori positive population is less than the H. pylori negative. And then interleukin 6 expression is highest. At the same time, interleukin 8/10 and tumor necrosis factor is lower in gastric cancer population. So discussion is, we would like to emphasize on the shifting pattern of ethnic incidence of gastric cancer in Malaysia, might be increasing incidence in Malay population. And H. pylori antigen detection pattern is not something that we have expected according to the literature. And the expression of interleukin 6 is upregulated in gastric cancer compared to control gastritis. DNF expression is increased, expression is decreased, and Indian ethnic mur is still there. We still should be found out. So that's all. Thank you so much.
Okay, thank you, Dr. Ted. Question from the floor? Anyone? All right, so just curious, well, when you see the interleukin 6 expression, does it correlate with the H. pylori infection?
It could be, Prof. We are doing the analysis now. And the remaining step that we are doing on is SNP, single nucleotide polymorphism. So it could be, and we are also trying to relate to the 3 major ethnic population and this SNP as well.
Right, and this IL-6 expression is from the blood samples or from the tumor tissue?
It's from the gastric cancer.
The tumor tissue?
Yes, the tumor tissue, Prof.
Ah, okay, interesting. Do you have the blood sample, because these are secreted cytokines, right?
Yes,
Do you
Prof.
see this in the blood of the patients?
Unfortunately, our data is, we screened that data from 2005 to 18, so it's not the fresh sample, Prof. It's from the gastrectomy tissue sample.
Okay.
Yeah.
Right. Okay. Yes.
Hi, Doctor. Nice to meet you. Doctor, do you have any information on their BMI status?
Unfortunately, no. We have traced the EMR with the permission from hospital and IMU. So in EMR, we do not have this BMI information, Prof. Yes.
Thank you.
Thank you. It's a very interesting question because IL-6 and inflammation and obesity. You should look into
Definitely, Prof. And if I may add a few seconds, these cytokines are involved in tumorigenesis as well as in suppressing the proto-oncogene. So I think dual role needs to be found out.
Yes.
Yeah.
Thank you. Thank
Thank
you
you
for the
very much,
interesting
Prof.
findings about interleukin 6.